So great. First of all, it's a great honor to be speaking after Yvette. I've known Yvette for, probably 20 years, maybe less, a bit, and worked with her pretty closely over the last 4 or 5 years where I was heading the PhD program at CMU, and she was, of course, as she said, the founder and the head of the University of Pittsburgh Computational Biology Department. It was very sad to see her leave Pittsburgh and move to Stony Brook, but as you have seen, the science at least is moving forward, so it was really great to be able to speak after her. There is some echo, no? I mean, it's... yeah, well, one is enough, I think.
If you get rid of this, maybe.
Okay, great. So I'm going to speak about spatial transcriptomics. Before I start, I wanted to say that a lot of this work was done by Hao, who is a postdoc in my group. And unfortunately for him, fortunately for me, he was not able to come because of visa issues. So I got to travel to Turkey and attend this great conference. Okay, so spatial transcriptomics, as many of you know, is, it's still, no? So maybe there's another one hiding here, Erchman? Or maybe I can speak loud enough that everybody can hear. I don't know, we'll see. Okay, so spatial transcriptomics is a way of looking at both the gene expression of cells and the context. I've been around long enough to see 4 waves, I think, of gene expression profiling technologies, microarrays, then we moved to bulk NGS, single cells, and now spatial transcriptomics. In my experience, once this such new technology comes in, it dominates everything else and quickly becomes the standard. So if I had to guess, although I'm not very good at predicting the future, this will be a technology that is going to take over the ability to do gene expression in the context. Now, there's many advantages for spatial versus single cell. The most important ones is that it allows us to look at not just at the different genes and cells, but also at interactions between them, networks, environment, which is very important for a lot of biological questions. In terms of technology, so the most famous sequencing-based single-cell technology is Visium, which is coming from 10x. There's lots of others. There's also imaging-based single-cell technologies. There are some advantages to sequencing-based technologies. But the major disadvantage right now of the technology, of the commercial technology, but most of the other ones, is the resolution. So spatial transcriptomics now usually... So spatial transcriptomics now usually works by profiling or sequencing genes on spots. And spots are pretty large and also far between. So usually we have between 10 and 25 cells per spot, and we never get single cell resolution for the data. There are a lot of interesting methods, including the convolution, copy number variation. You can do a few things, but of course, we don't get the single cell resolution, and in many cases, there's the distinguishability issues there. There's some new technologies that are now coming up, including 2 that I'm listing here, Sexscope and Stereosec, which are going into a much, much higher resolution, still using the spot idea, so basically still sequencing-based, but rather than having roughly 10 or 20 cells per spot, it's the other way around. We have now much, much more. We have hundreds of spots per cell, which is great because now we can basically, instead of trying to identify what is the different cells, try to basically combine the spots into one cell. But now the problem is the opposite. Before we had one spot for many cells, now we have one cell with many, many spots. And now the question is, how do we group spots together for an individual cell? The ability to move from a spot-based analysis to a cell-based analysis provides a lot of advantages. mainly, as I said, in the context of what we can and should do with spatial transcriptomics, including the ability to study cell-cell communication, right? If you work in a very large spot, it's very hard to know which cell is which in terms of the interactions. You can look at trajectories, you can look at microenvironments, and so forth. So what we try to do, or what we were hoping to develop, is a method that attempts at identifying which spots belongs to which cell. And you can do it in two different ways, and what we are proposing is to combine these two. The first is the standard imaging-based segmentation methods. Imaging-based segmentation starts by staining, sometimes with a nuclear stain like DAPI, sometimes with another marker for the membrane, and then everything within that stain is considered a specific cell. Another option is to look at the actual expression levels in these different spots and combine those that are very similar in their expression level, because they are likely to belong to the same cell. And what we are proposing is to use these two in combination and identify at a higher accuracy what are the different cells in these spatial outcomes. The underlying computational idea of SCS, of our method, has three major parts. The first part uses the actual cell segmentation based on the imaging, just on the imaging, as some sort of a self-supervised model, and I'll get to that in a second. The second part basically uses this supervised model to make predictions for each other spot, so spots that are not covered by the staining, about two things. One, whether they belong to a cell or whether they are part of the background, extracellular matrix, or basically not belonging to any cell. The second is, if they are part of a cell, what is the direction from this spot to the cell center or to the cell nucleus? And then the third part is, for all the spots that we determine to belong to a cell, we use a tracking algorithm or a flow tracking algorithm to group them together into a cell. Very briefly on the computational methodology. So, of course, you were working on imaging data. Even though most of the input is sequencing, you can't really not use deep neural networks. And in this case, we're using a pretty standard transformer model. There's two, I think, key ideas behind the way we are using the transformer model here that makes this method successful. The first is that this is a supervised model. And the question is, where do we get the supervised information? So where do we get the information on which spots are really a part of a cell and which are not? And what we decided to do is start by spots that we are very confident in. Specifically, if we have a very strong staining signal, for example, for the nucleus, we select all spots in the nucleus stained region as positive, and all spots that are far away from any nucleus stained region and have a very low intensity background as negative. And we train a model using these two sets initially. So this is the training data for the model. We don't need any label data. We use basically the staining itself, but we only use a subset of the spots for that. The second thing is, what is the input to the classifier? And it turns out that if you use the subcellular technology, a lot of people here used single cell data, and single cell data is, of course, very sparse. This data is even sparser. So usually in spots, you can have cases where you only have 3, 5, 6 reads per spot, which doesn't give you enough information for the classification. So instead, what we do in the model is we combine spots across a region. So we center the data on a specific spot, but the input is basically the vector, the gene expression vector from a lot of neighboring spots. In this case, we use 50, although we tried other numbers, and of course, the location information. And the output of the algorithm is basically two things. As I said, one is a prediction whether this spot belongs to is a part of a cell or not, and the second is the gradient, or the direction of that spot to the nucleus. Once we basically run, so we train the model, and then we run it on all the spots. Once we run it on all the spots, for each spot we get a binary prediction and the gradient, and then we use a tracking or a flow tracking algorithm to combine the cells and basically to combine the spots and segment them into a new cell. Okay, and of course, there's a lot of more details on the implementation in the paper. I want to talk a bit about validation, which is usually a very challenging thing in computational biology, and in this case, especially because we don't have ground truth. We don't know what are the cells and what are not. So how can we know that this technique that basically combines spots based on float tracking really identifies correct cell boundaries, or better cell boundaries than if we just use the imaging data itself? So for that, we use two data sets, very recent data sets. One is called, as I said, StereoSeq, and one is SexScope. StereoSeq is basically a brain data set with basically tens of millions of spots, tens of thousands of cells. And the other data set is StereoSeq, which is liver data set. Slightly less spots, but still quite a lot of spots, so roughly 500,000 spots. And thousands of cells. So these are the two data sets that we have. And now the question, and we applied basically our method to these two. And now the question is, how do you know if the cell, so of course our method gives us new cell boundaries or new cells, and any other segmentation method that is just based on imaging, which is what people usually do, will give you other cells. So how do we know which one is correct? So we did a lot of different validation analysis. The first one is basically looking at the agreement between data that is in the intersection of the two methods and data in which the two methods disagree. The idea is that if the two methods are good, then the intersection of the area they identified for each cell is probably correct. And now the question is, which one is better at identifying the rest? And what we looked at is the correlation in gene expression between the area that is shared between them and the area that they disagree in, which is what is shown here. Right? Oh, sorry. Right, so this is the area here. And what you see, and we looked at a lot of different methods, watershed, cell pose, deep cell, these are all very, very good cell segmentation methods, but only image-based. And you can see that in all cases, and specifically for StereoSeq, but also a little bit for SexScope, we get a much, much higher correlation between the intersection and the disagreement image for SCS compared to the imaging-based method. Of course, SCS uses sequencing, so it makes sense, but it's also a reasonable thing to assume that these are really more likely to be part of the same cell. But as I said, this is basically an indirect way because we don't have ground truth. There are cases where we have ground truth. In those cases, basically, the method we develop is useful, but there are other ways of doing it, but still, it's a good way to basically validate. And one of these methods is not a sequencing-based method, but rather an imaging-based method for the same goal of doing single-cell spatial transcriptomics. And this is called SegFish. Again, I won't go into that technology, but SegFish basically, or SegFish Plus in this case, profiles not all the genes, 10,000 genes, and of course it doesn't get you the sequence, so you need to use some other ways of identifying the imaging for the RNAs. But at least there, they actually did some manual annotation. Because you have the imaging data, you can actually manually, in some cases, annotate the cells. And we had ground truth to compare. And in that data, you can see actually, based on the ground truth, that SCS is doing much, much better than most of the other methods because it is able to combine not just the imaging, which is basically what other methods are using, but also the sequencing data. Okay, so this is good. We believe that SCS maybe identifies slightly better cell segmentations, but the question is, okay, what can you do with that? So the first thing is cell segmentation is really the first thing you do in any spatial experiment, so that's already a reasonable thing to do. But you can also derive a bit biological insights from these new predictions. For example, if you look at the cell size that the methods predict, because you can compare, you see that SCS identifies more realistic cell sizes, at least for StereoSeq, and SexScope. Again, the main difference here is that in StereoSeq, the staining was nucleus staining. So basically, you start with the nucleus and you expand with watershed or something else. And here it was membrane staining. So you already start with a better prediction of the cell, but still not completely accurate. The other thing you can do, and this is unique for sequencing, method that use sequencing, is identify cell types that you completely miss if you use the imaging-based method. Remember, we train using the imaging method, but the prediction is just based on the sequencing. So when we do that, we can identify completely new cells that were completely missed by the imaging because the intensity of the nucleus or the intensity of the membrane was lower than a detection threshold. And this is an example here where we have 2 new cells, or actually 3 new cells that are identified by our method, but are completely missed if you use, in this case, watershed, but this is true for other methods. And you can see here, this is not huge numbers, but there are cells that we identify and other methods not identify. And the most important thing about this is the identity of these type of cells. So it turns out that the ability to identify cells is not uniform. Cells that are on average larger can be identified by both methods. We identify them a bit more accurately, but other methods identify them as well. But smaller cells, which sometimes are of interest, sometimes they are more rare, are harder to identify by the staining methods and are much more, or not much more, but in some cases we identify many more of those by SCS. And again, these are some examples here. More generally, this is the correlation between the fractions of cells that we identify as new compared to other methods versus their size. And you can see that the larger the size, the more agreement there is between the two methods in terms of which cells we have. And the smaller the size, the less agreement or the more new cell types we can identify using sequencing and imaging rather than just sequencing. Last biological insight that you can gain is once you do that, because of the way we basically identify the cells, because we have the nucleus stain and the additional prediction based on sequencing, we can also divide the cell into the nucleus and cytoplasm and then do predictions based on that to identify which RNAs are more likely expressed in the nucleus and which RNAs are more likely expressed in the cytoplasm and derive some insights from that. So I'm almost out of time. I wanted to say that spatial transcriptomics, I think, is the future, or the present, but it's also going to be the future. So I think it's important to develop methods. There's a lot of exciting technologies in this space. Some of them are imaging-based, some of them are sequencing-based. And until now, at least the sequencing-based ones were more granular. So now we can actually look at much better resolutions with the subcellular organization. The method that we presented, at least we believe, identifies better cell segmentation. So if you are doing cell segmentation, which is usually what you have to do in the first place, you can just use that rather than use traditional based methods. And in some cases, this also identifies or provides some new biological insights. One last thing before I basically do the acknowledgement. I am still a professor at CMU. I've been there for many, many years. But now I also moved a year ago to work at a big pharmaceutical company called Sanofi. And there we work across the entire R&D value chain, including what I've showed you now, but a lot of other things. For example, I gained amazing new appreciation to what Yvette was talking about, the structure, once I moved to a pharma company. It's crazy how important this is there. So if anybody is interested in exploring pharma, I'm more than happy to talk to you. And the last thing is that I wanted to thank my students and my postdocs that worked on this, and of course the support of the NIH, especially through the HubMap and the Senate programs. And that's it. Thank you.